How does a single cell in your body actually know how old it is? We measure our lives in years, but on a molecular level, our bodies are keeping time in a much more complex way. New research is proposing that a fundamental biological clock might not be found in our DNA, but rather in the slow, steady transformation of our proteins over time.
So here's our roadmap for this explainer. First, we'll define what age really means from a biological standpoint. Then, we'll touch on the aging clock scientists already use. After that, we'll dive into this new hypothesis centered on something called disordered proteins, explore how this proposed clock might work, the challenges of actually reading it, and finally discuss what it could all mean for the future of healthy aging.
All right, first things first. Before we go any further, we need to be clear on what we mean by age. As it turns out, there are two very distinct ways to think about it.
On one hand, you have chronological age. That's straightforward. It's just the number of years that have passed since you were born. But on the other hand, there's biological age. And this is a measure of your body's functional state, its level of decline, and its risk for disease. This is why two people who are both chronologically 60 years old can have vastly different levels of health. Now, this concept of measuring biological age isn't new. Scientists have actually been working on this for a while, and they've developed several types of molecular clocks that give us a window into how our bodies are aging internally. Some of the more established methods include the epigenetic clock, which tracks specific chemical tags on our DNA. There's also the telomere clock, which measures the shortening of the protective caps on our chromosomes. Other approaches look at changes in our gut microbiome, the levels of inflammatory molecules in our system, or even the types of proteins present in our blood plasma. But the hypothesis we're focused on today suggests another kind of clock entirely. This one points to a specific class of proteins as a potential new way to track the passage of biological time.
These are called intrinsically disordered proteins, or IDPs. Now, you might think of proteins as having a precise, folded 3D shape, like a lock and key. IDPs, however, don't. They lack that stable structure, existing instead as a flexible, dynamic ensemble of different conformations.
And these IDPs are not rare. They account for about a third of all the proteins in our cells. Because of their inherent lack of structure, they're naturally prone to aggregate, to transition into a very stable, rigid form known as an amyloid. For a folded protein to do this, it first has to unfold. But IDPs already are unfolded, giving them a more direct path to aggregation. And this transition is a significant event. Once a protein locks into an amyloid structure, it is exceptionally stable. It becomes trapped in that state. You can think of it as a one-way street, creating a permanent molecular record that this change has occurred.
So it's this very tendency of IDPs to slowly but surely form these stable amyloid structures that forms the entire basis for the proposed IDP clock.
The central idea is this: the slow, persistent, and irreversible conversion of these flexible proteins into stable, aggregated amyloid structures could act as a kind of running counter, a direct measurement of a cell's biological age. Researchers suggest this process can happen in a few different ways. It might begin with just a single protein, a monomer, making that initial change in shape, a very slow event. Or it could start when small clusters of IDPs form, which then makes it easier for them to convert into a growing fiber. A third potential pathway involves dense cellular compartments, where IDPs can slowly transition from a more liquid-like state to a solid-like one, locking into that amyloid form.
And here's where the clock analogy becomes clear. Each time an IDP makes this transition into a stable amyloid form, and this is key, and it evades the cell's quality control systems, that single event represents one permanent, irreversible tick of this biological clock.
Of course, our cells have systems to counteract this. This network of defenses is called proteostasis. It includes things like molecular chaperones, which can prevent proteins from clumping together in the first place. The cell can also add chemical modifications to suppress aggregation. Even high concentrations of ATP, the cell's energy molecule, help keep proteins soluble. And for aggregates that do manage to form, disposal systems like autophagy are there to clear them out. All these systems are effectively working to slow down the ticking of the clock.
So the hypothesis is there, and it's compelling, but a clock isn't useful unless you can actually read the time. And this is where the practical and technical challenges come into play. There are three main hurdles. First, variability. The clock likely ticks at different rates in different cell types. A neuron doesn't age in the same way a skin cell does. Second is sample retrieval. It's very difficult to get samples from critical tissues, like the brain in a living organism. And finally, there's the issue of sensitivity.
In the early stages of aging, the quantities of amyloid are minuscule, so detecting them requires extremely sensitive technology.
To meet these challenges, scientists are looking at a few different methods. Non-invasive imaging, like PET scans, can detect larger aggregates, but that's typically at later stages. A direct tissue biopsy offers very high sensitivity for molecular details, but it is, of course, invasive. This has led to a great deal of interest in liquid biopsies, the idea that we could detect these changes through minimally invasive samples of blood or cerebral spinal fluid.
This really highlights a fundamental trade-off. With whole-body imaging, you get high resolution at the tissue level. You can see where the aggregates are. With a biopsy, you get very high molecular sensitivity. You can see exactly what the aggregates are. The goal is to find methods that bridge this gap.
But the real significance of developing a functional protein clock goes beyond just measuring age. It's about gaining a direct window into the fundamental health of our cells.
The proposal is that the IDP clock isn't really tracking years past, it's tracking health age. Its ticking serves as a direct readout of cellular stress and the declining effectiveness of the cell's protein maintenance systems. It's an indicator of functional decline.
So if it can be validated, why would an IDP clock matter so much? Well, it would give us a way to directly measure the health of our proteostasis network. It would also complement existing DNA-based clocks, providing a more complete picture of aging. The potential for reading it via liquid biopsy would make it highly practical. And it would give us a powerful tool to test if new interventions or therapies are actually working at the cellular level.
Let's look at a concrete example. The typical concentration of ATP in our cells is between 2 and 10 millimolar. We think of ATP as an energy source, but it also acts as a hydrotrope, which means it helps keep other molecules, including proteins, soluble. As we age, cellular ATP levels can decline. This weakens that protective effect, which could in turn increase the risk of aggregation and cause the protein clock to tick faster.
Ultimately, this line of research fits into a much larger shift in medicine. The goal is to move beyond simply addressing the symptoms of a disease after it has already appeared. The future, as this quote suggests, is to be able to treat people prophylactically, to address the root causes of age-related decline before they manifest.
This leaves us with a compelling question for the future. If we can truly learn to read our body's protein clocks, can we do more than just add years to our life? Could this knowledge allow us to proactively enhance our health span, the number of years we live in good, functional health?